Namaskar, welcome to PK's classes. Today we study about the monoclonal antibodies. Earlier we have studied about the polyclonal antibodies. So in polyclonal we call them as polyclonal because it is a mixed population of antibodies produced by multiple clones of B lymphocytes. As you know, antibodies are produced from the plasma B cells. So you can see a mixed population of antibodies, say antibody 1, antibody 2, antibody 3, each targeting specific epitope 1, 2, 3 in the same antigen. It is polyclonal. But when we prepare one antibody which can target a specific epitope, so there is single antibody species, then it is called monoclonal antibodies. So the monoclonal antibodies react with a specific epitope. Suppose these are various types of epitopes on the antigen. Suppose this is the antigen. So epitope 1, epitope 2, epitope 3, epitope 4. So it will bind to only, suppose say epitope 1, this epitope. And the population of antibodies which can target against this epitope only, that is called the monoclonal antibodies. So how do we prepare? So we will first inject the antigen against which we want to produce the antibody, and we will take the spleen, then we will harvest the plasma B lymphocytes, then we will hybridize it with a tumor cell, and that hybrid cell we call it as hybridoma. And this hybridoma, so one is your tumor cell, because of which there will be uncontrolled proliferation. We can produce a large number of hybridoma cells. Large number of hybridoma cells, and hence there will be production of monoclonal antibodies. The antibodies produced from these cells will be monoclonal antibodies. So we will again discuss how. So you can define monoclonal antibodies as these are the antibodies produced by a single clone of plasma B cells. They bind to single specific site or epitope on the antigen, and they are produced by the hybridoma technology. So they need the high level of skills, and production is slow, requires long production time, more than 6 months, and so it is produced in a very large scale. So you can see now, so we will first inject the antigen to the mouse, then we will collect the spleen cells from which we will harvest the plasma B cells, and then we will take the tumor cells or myeloma cells, and we take these because they can have uncontrolled proliferation. Now we will fuse the plasma B cell with the myeloma cell or tumor cell, and that cell is called as the hybrid cell. This is the hybrid cell. And we will select and grow these hybrid cells. Suppose we get these populations, then we will screen all of them for the desired antibody. Suppose we get this one. This one is acting against the specific, this is acting against the specific epitope in the desired antigen. Then we will select it and we will grow to produce large batches, and we will get the antibody which is called monoclonal antibody. They will act against the specific epitope in the desired antigen. So step by step we can say monoclonal antibody. First the antigen is prepared, then immunized to the animal, say mouse. Then the mouse develops the antibodies We'll isolate the spleen cells. From the spleen, we will harvest the plasma B cells and we'll fuse them with the myeloma. And the medium usually used is the hypoxanthine aminopterin thymidine. This HAT medium is used, incubated and screened to get the desired clone. Then we will expand it and purify it to get the desired antibody. Then we can store them. So when we take the large antigen immunogen that provides the multiple epitope polyclonal antibodies, but when we take the small peptide immunogen, it yields fewer restricted epitope polyclonal antibodies, and then we can isolate, fuse the B cells with the myeloma cells to get the hybridoma line and the desired monoclonal antibody we can produce. So what is the advantage with the monoclonal antibody? They will produce the homogeneous antibody population in contrast to the polyclonal antibody where there is a heterogeneous antibody population, and they are more specific, binds to the specific epitope in the antigen. The production is uniform and continuous, no batch to batch or lot variability is there. There is low cross reactivity. There is immortal supply because we have taken the myeloma cell, and there is high reproducibility because there is always, once the hybridoma is selected, then the production will be uniform. So high reproducibility there. So what are the disadvantages? It is expensive, time consuming, more than 6 months time is required, few host species options are there. All these are the disadvantages with the monoclonal antibodies. There are 4 types of monoclonal antibodies. The first one developed is the murine from mouse. Murine monoclonal antibodies developed by the hybridoma technology. Then this hybridoma technology is replaced with DNA recombinant technology. Then the second type is the chimeric. Then the third type, humanized monoclonal antibodies are developed, and finally now we have the human monoclonal antibodies. So we'll discuss one by one. Murine monoclonal antibody. So the hybridoma technology what we have discussed. This is the hybridoma technology, and using this technology we produce the murine monoclonal antibodies. Why we call it murine? Because it is produced from the mouse, plural mice. Murine monoclonal antibodies. Okay, and these murine monoclonal antibodies, the nomenclature, they end with omab. So you can see some examples. Detumumab, omab is there. Edrecolumab, abagomumab, and based on the nomenclature also they can be used against lymphoma, colorectal cancer, ovarian cancer, abagomumab. So the disadvantage with murine monoclonal antibody, they have a short half-life, they have limited penetration into tumor sites, so there is clinical failure. And the major disadvantage is they are immunogenic, so allergic reactions. So to reduce this immunogenicity or the allergic reactions, we have the second generation monoclonal antibodies. They are the chimeric antibodies. Chimera, you know, in Greek mythology, chimera is something which has a different body, different tail, the head of a different animal. So accordingly, here the murine variable region plus human constant region we will get the chimeric antibodies. As we know, this is the structure of the antibody, where this part up to this blue color. This blue color part is the constant region, and this upper color, this is the variable region. So variable region, this part will take that of murine, and this part will take that of human. Then we will have the monoclonal antibody, which is chimeric antibody, and it will approximately 65% human. So it reduces the immunogenicity. This is the advantage over the murine antibody, and they have increased half-life. So these are the two advantages. The nomenclature, they end with CIMAB, XIMAB. So you can see the example, AB6 CIMAB. It is anti-CD41 used to inhibit platelet aggregation, antiplatelet drug. Basilisk CIMAB, anti-CD25, prevent organ transplant rejection. Cetuximab, anti-epidermal growth factor receptor, so can be used in metastatic cancer, colorectal, head and neck cancer. Then the third group of monoclonal antibody is humanized monoclonal antibody, where we take the murine hypervariable region, or CDR, and here you can see, that means this part is the hypervariable region, this part is murine, and that is inserted into the human antibody, and the CDR we also call it as complementary determining region or hypervariable region. So by this technique we will get approximately 95% human, and the nomenclature, they end with JUMAB, JDUMAB, JUMAB. So bevacizumab, daclizumab, omalizumab, solanezumab, trastuzumab. Bevacizumab acts against vascular endothelial growth factor A, so used in metastatic cancer. Daclizumab acts as anti-CD25, prevents organ transplant rejection. Omalizumab is anti-immunoglobulin E, the FC region of immunoglobulin E, this FC region of immunoglobulin E, and this is used in allergic asthma. Solanezumab is anti-beta amyloid, so it is used in Alzheimer's disease AD, and trastuzumab acts against human epidermal growth factor receptor 2, HER2, used in breast cancer. Finally, the 100% human, the complete human monoclonal antibody, so nomenclature is human monoclonal antibody. Last one is humanized monoclonal antibody. So the final one, fourth type, is human monoclonal antibody, complete human antibody, and we can prepare this by transferring the human immunoglobulin gene into the murine genome, and we will get the transgenic mice, and these transgenic mice will be vaccinated with the desired antigen, and then we will prepare the monoclonal antibodies from this transgenic mice. So this is complete human antibody, so we call it as human monoclonal antibody. And the nomenclature, they end with umab, adalimumab, you can see umab, aducanumab, brazikumab. So adalimumab is anti-TNF alpha, used against rheumatoid arthritis. Aducanumab is anti-amyloid beta, it is used in Alzheimer's disease. Brazikumab is anti-interleukin 23, it is used in the Crohn's disease. So this is all about the monoclonal antibodies. Thank you.